post-transcriptional processing events. So you have the initial RNA that is produced, the heterogeneous RNA, needs to be spliced, it needs to be modified in all sorts of ways, and the processing steps are extremely well known. We talk about capping, we talk about cleavage at the 3' end and the addition of the poly-A tail. You'll remember that the poly-A tail is partly involved in defining the half-life, or the 3' end of the messenger RNA is associated with defining the half-life of that message in a cell. You have to remove the introns, of course. And all of these functions have to happen and are potentially sites for regulation of gene expression. Protein synthesis is a yucky slide, so I'll move on. Alison also briefly touched on different ways to analyze gene expression. We did in a tutorial in Wet Lab 2, we touched upon using real-time PCR for measuring gene expression. I was quite disappointed with some of the write-ups that you put in for that, because there were some very simple questions that got ignored. But I would suggest you go and review the tutorial again, because the evidence that I saw was that you hadn't grasped the basic idea that if you have a messenger RNA, if you purified messenger RNA, you can determine the relative abundance of specific messages in that by using real-time PCR, by converting the message into cDNA, using real-time PCR of a reference gene and an unknown, and comparing the CT values in real-time PCR to work out the messenger RNA's abundance. So I do suggest you review that tutorial that I had left on UTS Online. Dr. George, I've been working on Dr. George for quite some time, because as you probably started to appreciate, he just loves ABC transporters. He is supposed to cover translation, and the idea that the RNA is translated at the ribosomes, that different post-translational modifications of proteins occur, but I think he spent probably most of his time talking about ABC transporters. Anyway, from the exam point of view, we felt it quite important that you do appreciate the idea of understanding what is a proteome. The idea that you can analyze a proteome by making total cellular or tissue proteins, separating them by isoelectric focusing, and then SDS page to get a two-dimensional protein display of a cell or tissue's proteins, and then you can simply cut out the spots, digest them with trypsin, and do mass spectrometry. And through database searches, you can get an ID for that specific protein. So the idea of proteomes aims to identify and quantify the complete protein component in a cell or tissue, or specimen, or biopsy. So that could be a cancer, a tumour and a non-tumour, for example, at any one time. With the aim of trying to identify what proteins are present, the relative abundances of each of those proteins. Of course, the actual intensity of the spot on the gel reflects the abundance, how much of that protein is present, and allows you to compare normal and some other type, normal and diseased, different expression states or whatever. So Tony did go through the processes of sample isolation, 1 and 2D gels, mass spectrometry, and the fact that I put them in red shows it's probably in the exam. Anyway, the idea that you can use mass spectrometry to identify proteins by their time of Snapshot or Sequest or some kind of other search engine to take the mass spec data and investigate a protein database for similar proteins. Oh, this must be an ABC transporter. ABC transporters, well, what can I say about ABC transporters? I mean, you've heard it all before. The idea of looking at ABC transporters is to emphasize that essentially a protein structure, tertiary structure, the primary, secondary, tertiary structure of the protein is important for protein function. That proteins fold, many of the proteins have an order. The ABC transporters are one such class. They're important membrane pumps, and the idea that they are associated with ATP binding is an important, uses energy as part of that pumping process is an important concept. Tony tried to emphasize the idea of structure. I know he showed you lots of different beautiful pictures of his research models and stuff like this. It comes as no surprise, surely, that there will be some questions about ATP binding sites and ATP transporters. And Simpson has continued the theme of DNA makes RNA makes protein, and the final way we've tried to emphasize this is to try and get you to think about, well, if we know how DNA is transcribed into RNA is translated into a protein, we can use that knowledge and technology to our benefit in order to artificially express genes in bacteria, in eukaryotes, in viruses, and we can use that for benefits in terms of gene therapy, developing vaccines, transgenic animal technologies, and so on. So I have no doubt that you went through different structures of different bacterial expression systems. What do you need to express a gene in bacteria? What elements do you need to express in eukaryotic systems? I know talked about the idea of certainly for eukaryotic cells, if you have to get these products or these different DNA, RNA constructs into a cell, you have to transfect them into a cell, and talk about the different technologies used for transfection of mammalian cells. The gene gun, for example, is one that basically comes to mind. And then I have no doubt that Anne also talked about her research using different viral vector systems for expressing different types of things in eukaryotic cells. The retroviral and the viral and lentiviral systems are 3 different ones that were discussed. And then, of course, the most important thing is if you can artificially express eukaryotic genes in eukaryotic systems, well, how can we use this in an applied way? Gene therapy is, of course, Anne Simpson's expertise. So she will have talked about different diseases, the idea that you can artificially make constructs, you can alter gene expression in all sorts of ways to try and basically write disease states. Creation of transgenic animals is another fine example of using genetic engineering technologies, if I can call it that. Some of you might have seen in the news quite recently the idea that the Chinese now claim they engineered a cow to produce human breast milk. Doesn't appeal to me suckling another, right? But I have no doubt that there must be some money to be made in selling human breast milk. I'll leave you to think about that. But you have seen and heard about different other ways of genetic engineering, how it can be applied in a practical way. Does anybody have any questions? Anybody wants to ask why some are in red and some weren't? Yes, what is the structure of the question? The questions can be in parts, so there are like A, B, and C. Sometimes you're asked to draw a picture, sometimes you're just asked to write something, define something. But do make sure you answer all the parts and make sure you identify which part you are answering. And just do put your name on the front of the book. Might seem sensible, but any other questions?
Why were they white and red?
Look, I know some of you have got to be